Welcome to Let's Get to the Marks, going through an AS biology question on DNA. So this is good for your revision on DNA. It's a walkthrough, so pause the video, read the question, have a go, then unpause and watch the walkthrough, and basically I go through the answers. So the question that we're starting with here is 1.1. The nucleus and the chloroplast of a plant cell, they both contain DNA. Give 3 ways in which the DNA in a chloroplast is different to the DNA in a nucleus. 3 marks. Here we go. Okay, chloroplast contains circular DNA. And this is due to the fact that chloroplasts evolved from prokaryote-like creatures that ended up moving into our ancestral single-cell eukaryotic cells. And you can look up that in the description with endosymbiosis if you're interested. Also, the DNA in chloroplasts is not joint or wound around histones, whereas the DNA in the nucleus is bound to histones, and you learn a little bit about that with acetylation and methylation as well. The DNA in chloroplast is obviously shorter in length because it's only got the instructions for the chloroplast rather than all the other functions in the cell, and so it only contains a few genes or a lot less genes than the DNA in the nucleus. Also, the DNA in chloroplast does not contain introns. Okay, 1.2. Some DNA nucleotides have the organic base thymine, but RNA nucleotides do not have thymine. RNA nucleotides have uracil instead of thymine. Give one other difference between the structure of a DNA nucleotide and the structure of an RNA nucleotide. So here I'm drawing out the structure of your DNA nucleotide. You should be able to remember the 3 main parts of a DNA nucleotide. You have your base, nitrogenous base, which can be adenine, thymine, guanine, or cytosine. You have your deoxyribose, which is a pentose sugar, and you have a phosphate group. So there's your DNA nucleotide. Well, what's the difference with an RNA nucleotide? Well, the RNA nucleotide, as we've been told in the question, so we don't get a mark for this, has uracil instead of thymine as one of its bases. They both have a phosphate group, so we can't put that as an answer. And they both have a pentose sugar, but there is a difference in the pentose sugar. The RNA nucleotide has ribose sugar, whereas the DNA nucleotide has deoxyribose. So our answer, and only answer for this question, is that the RNA nucleotide contains a ribose sugar, whereas the DNA nucleotide contains a deoxyribose sugar. And that's the only possible answer for this question. You could get a question on the similarities, so make sure you can see the similarities. You've got the AGC for the nitrogenous bases. You've got the phosphate group. They both contain a pentose sugar. Those are lots of similarities as well. So make sure you know those off by heart as well. Okay, for this part of the question, 1.3, figure 1 shows a tRNA, transfer RNA molecule or carrier molecule for amino acids. 1.3, name the structures W and X in figure 1. So starting with X, as it's the easier one, you can probably tell what do we call the little code, the little triplet code or codon that you get on a tRNA molecule. We call it the anticodon, and that's because it's complementary and binds to the codon, which is found on mRNA. So you would have like GAG would bind to this CUC, codon and anticodon. So the answer for X is anticodon. Now, W, what is it that tRNA molecules are carrying, people? What is being carried by a tRNA molecule? It's an amino acid. So this must be the amino acid binding site within the tRNA molecule where it picks the amino acid up. And you need both of these to get yourself 1 mark. So that was a little bit tricky. Let's move on to the final part of this question for this video. And it's 1.4. Not all mutations in the nucleotide sequence of a gene cause a change in the structure of the polypeptide. Give 2 reasons why a mutation might not cause a change in the structure of a polypeptide. It's worth 2 marks. Pause the video. Have a go. Okay, so think about it. Why might you get a mutation, a change in the code of a gene that doesn't change the structure of the polypeptide? It doesn't affect the tertiary structure, essentially, of a polypeptide. Well, let's look at this example. UGU codes for cysteine, okay, which is one of the 20 essential amino acids, but also UGC codes for cysteine. So you've got 2 options there. And that means if our final little U, our final uracil changes to cysteine, then essentially, what the hell am I waffling about? Anyway, yeah. This is basically pointing out that DNA is degenerate and that each amino acid has more than one triplet code. Okay, so, or yeah, the same amino acid can be coded for by more than one triplet code. This is when you get those substitutions, which can result in silent mutations where you don't actually change the polypeptide structure and therefore the tertiary structure and the function of the protein. Okay, so like if a mutation changed the U to a C, it would not affect the amino acid being coded for. I don't know why my phone's beeping away. Okay, there's also another reason why a mutation wouldn't result in the change in a polypeptide structure, because the mutation could occur in the intron section of the DNA. And of course, the intron is the non-coding section of the DNA. You're talking about the regulatory genes or the junk DNA on the sides of the actual structural genes. Remember, the structural genes, the exons, they code specifically for the polypeptide. So unless you have a mutation in those exons, you're not going to get a change in the function of a polypeptide. So there's your two answers for this one. The mutation could occur in the introns, or the fact that DNA is degenerate and you could get more than one triplet code coding for the same amino acid means it will not affect the primary structure and tertiary structure of the protein. All right, guys, thanks for watching the video. Let's get to the marks.
You're still here? It's over.